Hello everyone, my name is Katie Atherton. I have been working with Professor Pashuk in the Department of Bioengineering, and we have been working on identifying peptides with cell-specific degradation. So to give some background, peptides are widely used in hydrogels and other biomaterials because they are relatively easy to synthesize, and they can greatly enhance the biological properties of biomaterials. They help modulate cell adhesion and signaling, and they cross-link the polymer networks of the biomaterials. Additionally, they are natural substrates for cell-secreted proteases, which makes them ideal for enabling the biomaterial matrices to be modified by the cells they contain. So there has been significant work done to understand how biomaterials influence cells, but we know that cells can also influence biomaterials. Additionally, peptide degradation has been studied by incubating peptides with individual proteases, but we know that protease activity varies greatly depending on cell type. Additionally, peptides can be degraded by membrane-type proteases, meaning proteases that are attached to the cell membranes, and they can also be degraded by soluble proteases, meaning proteases that have been released into the media surrounding the cells. So we speculated that there were peptide sequences that are cleaved only by some cell types and not by other cell types, and that we can use membrane-type proteases to limit peptide degradation to the area immediately surrounding the cell. So identifying this cell-specific and membrane-specific peptide degradation will help enable the design of hydrogels containing multiple cell types, but having controlled cell-specific environments. So the objectives of our study were to, 1, identify peptides that are degraded by one cell type and not by others, and 2, identify peptides that are degraded by membrane-type proteases and not by soluble proteases. So to do this, we incubated peptides and we quantified their degradation with LC-MS. We used 6 cell types: human umbilical vein endothelial cells, or HUVECs, human mesenchymal stem cells, or HMSCs, and 4 types of macrophages. So we cultured the cells in media for 24 hours, and then we separated the conditioned media from the cells, and we replenished the cells with fresh media. We then added the peptides to both the conditioned media and the fresh media containing the cells, and we incubated them for 24 hours. And then we quantified this peptide degradation using LC-MS. So we divided the amount of peptide present at the 24-hour time point by the amount of peptide present at the 0-hour time point. So peptides with a degradation number closer to 1 indicates that there was less degradation going on, and peptides with a number closer to 0 indicates that there was significant degradation that occurred. So of 18 peptides that we looked at, we identified 4 that were degraded significantly by HUVECs or HMSCs, but not by the macrophages. And we identified 3 that were degraded only by the condition-containing cells and not by the conditioned media, meaning that they would be degraded by membrane-type proteases. But overall, we noticed that degradation varied greatly on cell type and condition. HUVECs and HMSCs tended to result in more degradation than the macrophages, and the cell condition tended to result in more degradation than the media condition. The poster goes into more detail, looking closer at the specific numbers of the peptides that were degraded by HUVECs and HMSCs, and looking at the peptides that were degraded by membrane-type proteases, but not by soluble proteases. So to sum up our work, we developed a methodology to identify and quantify peptide degradation by membrane-type proteases on specific cell types. So future work would include creating large peptide libraries and further testing, using peptides modified by a photolabile linker to make hydrogels, and then quantifying the peptide degraded, and then using microbiology technology to measure the degradation gradient of hydrogels, cross-linked with the peptides that we made. I hope you enjoyed listening. Thank you so much.